Cek, cek. Bismillahirrahmanirrahim. The Honorable, the Dean of Faculty of Marine Science and Fisheries, Hasanuddin University, in this time is present by Vice Dean for Partnership and Alumni. Our esteemed speakers, Mr. Japareng bin Lalung, PhD, from the School of Industrial Technology, University Science Malaysia. Our distinguished guests and lecturers from Faculty of Marine Science and Fisheries, and all dear participants. Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Good afternoon, and our warmest welcome to the Blue-Green Algae General Lecture with the theme Toxin-Producing Cyanobacteria from Global Threat to Aquatic Ecosystems and Human Health to Potential Solutions in Biodiesel, Plastic Degradation, and Skincare. Organized by the Aquatic Resource Management Program, Faculty of Marine Science and Fisheries, Hasanuddin University. My name is Sabika, and I am honored to serve you as your Master of Ceremonies for today's event. Today, to begin today's sessions, let us open with the welcoming remarks by the Dean of Faculty of Marine Science and Fisheries, and in this time will be presented by the Vice Dean for Partnership and Alumni, Professor Mukti Zainuddin, PhD.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, distinguished guests, respective college lecturers, dear students and all participants. On behalf of Faculty of Marine Science and Fisheries, Hasanuddin University, it's a great pleasure to welcome all of you to today's special lectures. We are deeply honored to host this special event and to have Professor Jeb from the School of Industrial Technology, University Science Malaysia with us. His lectures entitled The Toxin Producing Cyanobacteria from Global Threat to Aquatic Ecosystem and Human Health to Potential Solution in Biodiesel, Plastic Degradation, and Skincare. I hope these lectures offers excellent opportunity for us to gain new insight into blue-green algae from an internationally recognized researcher. In these special lectures, I encourage to all of you, to all participants, to participate actively and ask questions as many as possible and make the most of valuable this opportunity to learn and exchange ideas. Finally, I would like to express my sincere appreciation to Professor Jeb for accepting our invitation and sharing his expertise to our academic community. Thank you very much, Professor Jeb. let's open officially this lecture by saying Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for Professor Mukti Zainuddin for the opening. Before we proceed to the main part of the event today, we would like to introduce our speakers. Therefore, with great respect, we would like to invite Dr. Rernat Elmi Nurhaida Zainuddin to introduce our speakers for today.
Sure. Oh ya, saya ngomong bahasa Indonesia aja ya, karena ini kebetulan adik-adik semua ini supaya lebih paham ya, apa itu cyanobacteria. Ya, boleh ya. Jadi beliau ini adalah peneliti dari Universitas Sains Malaysia dalam bidang mikroalga cyanobacteria. Cyanobacteria itu adalah kelompok mikroalga yang berfotosintesa tetapi bersifat seperti bakteri. Kenapa dinamakan cyanobacteria? Karena ada dalam kelompok Cyanophyceae dan masuk dalam kelompok prokariot bakteri. Jadi itulah keistimewaan dari jenis mikroalga ini adalah kelompok bakteri yang bisa berfotosintesa. Itu keunikannya. Nah, beliau ini expert dalam penggunaannya, aplikasi cyanobacteria, dan bagaimana juga bisa mengeksplor di alam. Jadi di alam nanti itu cyanobacteria kita bisa temukan seperti apa, kemudian kita manfaatkan seperti apa, itu nanti akan beliau jelaskan. Mungkin kalau Anda mau bertanya nanti tidak bisa dalam bahasa Inggris, boleh bahasa Indonesia. Beliau ini orang sini, Sulawesi Selatan, orang Enrekang. Tetapi lahir, besar, sekolah di luar negeri semua. Jadi maklum kalau bahasa Indonesianya agak ini ya, berdialek-dialek Inggris atau Inggrisnya ada juga terikut, itu harus dipahami juga. Tapi beliau akan mempresentasikan, diusahakan dalam bahasa Inggris supaya kalian juga belajar untuk memahami dalam bahasa Inggris. Nggak apa-apa kan? Oke? Ya. Please, you can take your time now. Thank you.
Okay. Terima kasih ya. Okay. And first of all, thank you so much for accepting my self-invitation to come here. So I think as what Prof. Elmi said, my parents are from Indonesia. So my father and my mother was born in Indonesia, but they moved to Malaysia. So I was born in Malaysia, but still I think I'm 100% Indonesian by blood. But yeah, but I am Malaysian now. So I wonder if the slides is going to be ready. I have long slides to present, about 200, but bear with me, it's going to be quite quick. And the fact that I cannot control myself, so I'm just going to give a signal to the technical guy. I don't know what's his name.
I will give you the sign.
Oh, all right.
I helped you as a moderator today.
Oh, right, okay.
Moderate this, yeah.
Okay.
Sorry. 45, yeah.
Okay, I've got 45 minutes, yeah.
Yes.
Okay. So it's going to be a long lecture. So because this is, I think, mostly undergraduate, so it's going to be quite basic. I'm going to talk about some fundamental things and hopefully focus into my research. And hopefully one day we can work together, and also I think to the lecturers and also the students, I hope we can work together in the future. So the title is Toxin Producing Cyanobacteria from Global Threat to Aquatic Ecosystems and Human Health to Potential Solution to Biodiesel, Plastic Degradation and Skincare. So yeah, I'm trying to wrap up of everything that I've done for the past, I think more than 10 years being a docent. Yep. So if you can go to the next one, please. All right, thank you. I think we started by saying thank you to UNHAS, especially Prof. Elmi, Prof. Mukti, and Prof. Sri Wahyuni. And if you can go to the next one. I don't know Prof. Sri Wahyuni, maybe she's not here today. Oh, okay, thank you. Sorry, I forgot about that. Okay, and okay, Prof. Wilma also. Sorry. Well, let's pray together. So this is also the people who work in the lab, to postdocs, current and former students. I think about 80, all of us together. I think I missed some of them, but I want to say thank you for their contribution, because this is going to cover everything that I've done. If you go next one, please. So a bit of my background. My name is Japareng Lalung. I think it's a very Bugis name. And I work as an associate professor at the School of Industrial Technology, University of Science Malaysia. And I'm also involved in something called Regional Center for Expertise. This is actually a unit established by UNESCO under United Nations University. I am now holding that post in the Center for Global Sustainability Studies in University of Science Malaysia. So we are very well aware about the sustainability agenda of the university. And I teach environmental science, environmental management, microbiology, forensic, solid waste management, and other subjects. If you want to ask any questions, you can just raise up your hands, and you can also ask me in Bahasa at any time, you know. I like it when people ask me questions. I might give you some present as well. Who knows? If you want to go the next one, please. So this is my background. I studied in University Malaya, and I went to Oxford University, and also I went to Leeds University in the UK. If you want to go to the next one, please. Okay. So I also, before joining academic, I used to work with the private company, one of which was being a manager in the biggest water treatment company in Kuala Lumpur. But then I've decided that being academics is actually much, I would say, easier. So I left private sector and joined academics. If you want to go to the next one, please. I'll just give you a signal like this, yeah? And this is my research interest: water, cyanobacteria, blue-green algae, biodiesel, and bioremediation. Hopefully we can venture into this afterwards. If next one. Yeah, I wrote a book. I also involved in Malaysian Antarctic research program. So every now and then I'll be like sent to Antarctica and do some samplings. And this is one of the book I wrote about the experience that I had. I can talk forever about Antarctic research, but today I think we're going to focus on cyanobacteria. Next one, please. And also I was accidentally involved in waste-to-energy, and this is due to the visit that I did in China in 2024, but maybe you're not going to talk about this today. Next one, please. Yeah, this is our recent focus. It's on scytonamin. This is actually a pigment which is used by cyanobacteria to block sunlight, a UV ray from killing them. And you can use this for your skincare, and this is very expensive. So this is what we're doing now. I think almost like 80% of our lab is doing research on this. This is very expensive. You could look at that. I think it's about $65,000 per gram. So if you want to do research, this is what I Prof. Matma. About potential research, I think Indonesia is going to have a lot of species that can do this, because biodiversity is Indonesia is one of the richest in the world. Can I go to the next one, please? Yeah, I also currently consulting with my former company, and yeah, because I basically know what their problem is, because I used to be working with them, and now we become the consultant, and also on oil producing algae. If you want to go to the next one. This is my research stream in 2026, but I think 7 of them graduating this year. So the number is going to be reduced because 1, 2, 3, 4, 5, 6, 7, 8 of them finished this study this year in October. But I think we're going to get the new ones because our turnover is actually quite high. We have people coming in and out. Next one, please. And this is our current research. Actually, 2 things. One is producing the skincare, and the other one is on breaking down plastic. And also we are heavily involved with education for sustainable development. So we wrote books on education for sustainable development. We did it in Penang, and we're planning to extend to Malaysia this year, and hopefully next year to Southeast Asia. And hopefully Indonesia can be one of them. If you, next one, please. And this is our collaborator. I think I'm just gonna go, yeah, Malaysia, UPM, International Islamic University, and also with the police in Malaysia. If you're gonna, next one, please. We also have international collaboration, GBU, Gujarat in India, Chile, Canterbury in New Zealand, Amity in India, Al-Farabi in Kazakhstan, and I think another one is something like Walisono, somewhere in Sumatra, and Andalas. All right, sorry. The next one, please. Yeah, we work together with Malaysian Antarctic Research Program, with Korea, Sejahtera Forest, Shia Kuala, and Gajah Mada, and also with British Antarctic Survey. If you go to the next one, please. Who's controlling? Okay, and Shenzhen Institute of Advanced Technology, University of Vienna, Brawijaya, and Satbayev University in Kazakhstan. So this is, hopefully I put that empty one, and that's actually, I hope Unhas will be one of them in the future. Next one, please. So I want to introduce about Penang. Penang, next one, is actually a UNESCO heritage site. The city itself is a UNESCO heritage site for culture. If you can click, please. I don't know if I can I just handle the mic? Is it possible or not? Is it possible? No? Yeah, if you have, if not, it's fine, yeah. Yeah, because it's a UNESCO heritage site both for culture and also the surrounding, that is forest, is a UNESCO biosphere. So I think Malaysia is very good in claiming things, as you well known. So we wanted to make story out of everything that we have. So one of them is a UNESCO heritage site for culture and also for the forest around. If you can go for the next one. Okay, this is University Science Malaysia. It's located in Penang, which is the northwest of Peninsula Malaysia. If you go to the next one, because this is like 200 slides, that's why I wanted to control myself, because I can skip some of them. So we have 9 main campus. I think it's about 30,000 students altogether, and that is one of our research study center. That is the main campus up there. We have a lot of forests, just like in Unhas, because we have a lot of nice forest. And the one below is the Center for Marine Studies. If you can go to the next one. Yeah, we have a lot of campaign on sustainability. We cannot use single plastic, we cannot use polystyrene, and now I'm involved in the committee who is actually counting on food waste. We do not want to waste food in the university. And, okay, it's okay, okay, thank you, yeah. And if you press the next one, please. I think there are like 3 times to press, yeah. And in terms of global impact ranking and QS ranking, we are ranked this year, 2026. For QS ranking, we are ranked 134 in the world, and for global impact ranking, we are ranked 5th in the world and top in Asia. But we wanted to be number 1, to be honest. So we are working very, very hard to be on top, and we had a strategic plan, and hopefully it's going to work. Okay, but maybe not. We never know. But we are looking for. We are very we are being optimistic. If you want to press the next one. Okay, in our school we have an environmental technology program where I'm sitting, and the next one. We have food technology program. The next one, bioresource technology, and the next one is bioprocess engineering technology. This is actually by one school with 56 or 54 lecturers altogether. If you can press the next one. And we also have masters on environmental science for sustainability. This is 1 year, and we've got a lot of students from Indonesia and mostly China now. And I think for this course about 30 people, and I think about 80% international students and 20% local Malaysian. If you press the next one. Okay, the introduction, where did it begin? So I'm going to talk about the science that I'm doing. So this is Putrajaya Lake, where I first started my career, when I first started seeing cyanobacteria a long time ago. If you want to see the next one. Okay, so cyanobacteria, and I think this will be very good for undergraduate. I cannot read this actually, but never mind, I'm going to try. So it's actually a group of I think Prof. Elmi already said this. This is actually photosynthetic, and they are basically everywhere, and basically almost the only limiting factor that they have is the sun, simply because they can scavenge phosphorus, very little amount of phosphorus, and they can fix nitrogen from the atmosphere, so they can grow easily. So if you look at the water bodies, I think if you put this outside and leave it there for a long time, it's going to turn green, and that's cyanobacteria. And one of our research is on why do they live on plastic, because they don't use carbon. They're photosynthetic, that means they're doing photosynthesis. They are not, they do not need the carbon, and the water is clean, it's not even dirty. Why are they still surviving? And very often people think cyanobacteria is actually linked with pollution. I don't think so, simply because even if the water is very clean, they can still grow, simply because they don't need fertilizer, they can fix nitrogen. So if the water is clean, the nitrogen fixing strands is going to grow because they can just fix it from the atmosphere. So almost basically what you need is lights. And our recent studies prove that they can actually even take phosphorus from plastic, and now you have phosphorus and you have nitrous, and you can grow. That's why you have quite a substantial amount of blue-green algae blooming. And this is not necessarily bad, you know, because actually 60% of our oxygen is not produced in the forest, it's actually produced in the sea by cyanobacteria. So they're very important for us, otherwise we don't have enough oxygen. But I think most of the time, I think understanding about cyanobacteria is crucial, and it's also very important because we always think them as our enemy. But actually they're not, they can be very nice. Very often I tell my students, do you want your forest to be green? What happens if you leave the land? It's going to turn green like the Amazon and also the rainforest in Indonesia, because we have enough lights. And you want it to be green, but then you water, when your water turns green, you look at it as a negative thing. But in reality, they're also doing photosynthesis, and they're producing oxygen. And that's why it's almost impossible. People expect the water to be pristine, clear, no green thing. But like the forest, it's going to turn green when you leave it alone, and the same goes to your water in the tropics. And if we didn't understand this concept, it's going to make our life very difficult. If every time we wanted to kill them, knowing their nature is like that, it's going to be very difficult. So instead of like arguing and looking at them as enemy, what we are trying to do now is actually to take advantage of them. For example, if they're bloom forming, if they can produce oil, that means they can produce oil faster. If they can produce the UV protection, they will produce it faster. So this is the thing that we are trying to do in our lab. But maybe you can go for the next one, because otherwise I'm going to use up all my time. I have 20 more minutes, 30 maybe. So this is overall what it is, unicellular, but I think we can skip this. This is how they look like. If you want to go to the next one, please. Yeah, this is specialized things about cyanobacteria. They have archinates. Basically, it's like spora, and they can live for a long time. You can have them even for 100 years, you know. So they can, and when the conditions is right, they're going to grow. So this is why they survive very well. Next one, please. And what is this? I don't know. Hormogonia. This hormogonia is actually small filaments. They can disperse easily. So that's why now they can leave, and then they can move easily to different places. They can disperse easily. If you go to the next one, please. And this is this, never mind. It's vesicle. I cannot read this, actually quite difficult. All right. Okay. Nitrogen fixation. So nitrogen fixation is actually heterocyst, but I don't think this is heterocyst. Maybe you can go to the next slides. Oh, never mind. Yeah, it doesn't say. So it can also fix nitrogen, as I said. Yeah, so environmental impact, when algal bloom happen, what happened is that, do you know, I want to ask you these questions. If you can answer these questions, I'm going to give you a present. If the water turns green like that, do you expect the oxygen to be a lot or little? Yes? Why? Can yeah.
Karena oksigen itu digunakan oleh
Okay. If the forest is green, is there going to be a lot of oxygen or no? Kalau hutan hijau, banyak oksigen atau kurang? Banyak. Kalau air hijau, oksigen banyak atau kurang? Profesor mesti for sustainable development. If you go for the next one, I just give you example what we have done in University of Science Malaysia. Maybe you can go to the next one. I don't want to talk about this background when it was established. Next one. Habis sudah. Okay. Maybe just a bit script. You can just go through, like skip. This is what we're doing. Next. So I had this team. We have like students and next one, until you see. So this is what we're doing in Malaysia. These are all involved community project that we've done. This is headed by my team, and we've got awards actually in Okayama last year. Next one. Go to the next one. We keep on planting with everybody in the world, basically. Yeah. And I can talk about also about palm oil sustainability, which is relevant to Indonesia. We're promoting palm oil a lot. Next one, because I want one. Next one. Okay, we've got the awards. This is the book that we're writing. And next one. Last year we won awards in Japan, Okayama. Next one. Just press next until the response slides I want to show. Yeah, this is this year, 26 this year, and that's it. Next one. Yes, thank you very much. Because this is about. I want to show picture about my Antarctic research while we have question and answer. Thank you. Thank you so much.
Okay, let's give applause to Assistant Professor Jeb here with his very interesting and opening eye presentations. And before we go for the Q&A sessions, I would like to ask for the photo session first. Is it possible? So maybe the photographer will lead us how to, where will be. Ya.
Ayo maju ke depan saja kita ambil ke sana. Oh sini boleh juga. Ya.
Bagaimana tampilannya?
Kita bagian depan, kita di depan. Ya.
Oke, saya rasa saya juga tidak perlu mengulang terlalu banyak ya. Semoga penjelasan presentasi tadi yang sudah dijabarkan oleh Dr. Jeb sangat menarik, dan saya harap kalian bukan hanya melihat itu menarik, tapi memberi selain membuka wawasan, pasti memberikan spirit bahwa melihat banyak sekali potensi yang bisa dikerjakan hanya dengan berbicara cyanobacteria. Ya, bisa banyak hal yang dilakukan dari itu. Dan silahkan kalau ada mungkin yang ingin ditanyakan lebih lanjut, boleh ditanyakan ke beliau. Saya buka sesi pertama. Silakan Mulki, Prof. Nita. Ya, Prof. Silakan, Prof.
Langsung? Okay, thank you. Thank you, Dr. Jeb. It's very interesting talk. So I've been working with cyanobacteria since 2011. I work work with one of the professor at Reno University, how to search the cyanobacteria for biofuel, microalgae for biofuel, and continue work with one of the professor in Uppsala University, and also work with the microalgae to search the biofuel, the potential of biofuel. Before that, 2010, I worked with Prof. John Birdell, also working using microalgae, especially for cyanobacteria, for mitigate the CO2 climate change. So my work with my professor is actually is very interesting. As you said that the cyanobacteria very high potential as a biodiesel or biofuel. But the problem is the technology to extract lipid or whatever H2 from the microalgae is really costly, really high cost. So even we can produce the big scale of the microalgae with the small area of land, because we can use the with the vertical thing, so it's the very potential. But the problem is till now, even in America or in Europe, so we still have struggling to convert the lipid of the lipid from the microalgae or from cyanobacteria to be the potential biofuel. I just wondering the last research, your research about cyanobacteria, what is the main challenge for you to search the cyanobacteria for the biofuel? This is the first question. The second question is, I really interesting about, you mentioned that related to the what you call palm field, right? Kelapa sawit. Ya, artinya perkebunan sawit. So you mentioned that the log of the sawit can protect cyanobacteria for growth. So do you, but we really need some, what you call, some bacteria or maybe some cyanobacteria that can be used to, to convert the waste of the kelapa sawit, the palm plant, to more have the value added. Do you have any research regarding this kind of topic? Why I ask that? Because at the moment in Indonesia we have a huge grant. That grant came from the all private company, semua perkebunan, like a CSR of all the private company. They provide the grant to do the research to maximize everything, to find the technology, the innovation, to maximize the everything about the palm and cocoa and also coconut kelapa. So maybe you can share with me, do you have any work related to the using cyanobacteria to give the value added of the palm waste? Maybe there is only 2 questions. Thank you.
Thank you, Prof. Nita. And
Thank
maybe the next question first. Can I?
I don't know if I can remember. Can I answer?
Oh, okay, you can ask. Okay, you can answer, sorry.
Yeah, okay, first question, actually very valid question. One of the biggest problem with converting microalgae or cyanobacteria into algal oil or biodiesel is the cost. For as long as diesel is cheap, I don't think it's going to work. That's my opinion. It's very blunt. A lot of people disagree with me. It's almost impossible because oil is still cheap. Like 10,000 rupiah per liter, you cannot produce that alga oil that cheap, you know. It's not that cheap, but you want to produce alga oil, that will be a big challenge. Then the second challenge is the hampas. You know hampas? Yeah, you're going to be tons and tons of them, the sisa. So we have this issue. So what we did was we're trying to do zero waste. We feed the sisa into ayam. You know what happened to our ayam? Mati. So and then we realized silap lah, because ayam is makan kajagung, so we should feed it to kambing or cow, sapi. But we haven't done that experiment simply because even the oil itself is not going to be worth it. But in the future, if we were going to do that, we're going to feed it to sapi. Tapi sapi is like long, kalau mati sapi rugi banyak kan. So ayam lagi murah. So that's why we started with ayam. But I think that will be the biggest two challenge. One is the cost. The second is the sisa. Yeah, in terms of space, it's going to be better than palm oil. Sebab you use less, you know, especially the bloom forming, you're going to produce a lot. Yeah, theoretically it's possible, but I think, in my opinion, I'm sure a lot of people disagree with me, for as long as petroleum is there, I don't think it's going to work. But once the petroleum is gone, then you have no choice. Yep, that's my question. The second part is about the palm oil. Actually, what we're doing with, I don't really have exactly research that you are asking for, but what I can confirm is that we can use the palm oil trunk to actually control cyanobacteria. Just like any lignin derivatives, anything that can produce lignin, it can actually control cyanobacteria. So what we can do, what we did was we put palm oil trunk into the water and cyanobacteria stops growing. That's what we did. And you basically, all you need to do is to dry it. You can claim that you're doing treatment, but actually you don't need to do anything. Just put it there, it's going to be okay. I hope that answers your question, but anything else I didn't really do. Thank you, Prof. Yeah, and I think I need to give you some. Professor must be a lot to give kan, anak bagi semua lah. So you're gonna have a complete 18 set, but Professor, yeah. But students satu saja kan, yeah. Okay.
Okay, thank you for the answers. Next will be from Mulki. You can have your questions.
Thank you very much. I'm interesting with the spirulina, and then I know the sorry.
Boleh bahasa boleh. Saya pandai.
And I know the some cyanobacteria is the toxic. So can I determine whether cyanobacteria is toxic or non-toxic? Also based on the color or apa tadi ya? Morphology, and how can I apa ya? Yes, identify it.
Yeah, is there any way to identify the cyanobacteria if they are toxic?
Thank you so much. I think that's a very good question. So saya bagi dua. Tapi belum profesor lagi kan, so tak boleh bagi semua. Okay, what's your name by the way?
Mulki.
Okay, that's a very good question. So toxin producing is not species specific; it is gene specific. So that means the same species can produce or not produce the toxin. So that's why making it quite difficult to determine. So it's gene specific, not species specific. So same morphology, some of them produce, some of them not. So what we do is, the first thing, if you wanted to know whether it can produce toxin or not, you can actually see the gene which encode for secondary metabolite, the PS gene and the polyketetes PKS gene. Yep. Yes. Takut sekarang ada profesor di belakang. So what we can do, you can actually look at the gene. You can use DNA. If it has that gene, then it can produce toxin. It can produce secondary metabolite. If it can produce secondary metabolite, it can produce toxin. So how do you know it can produce toxin? You screen for the gene encoding for the toxin, not morphology. So the same toxin can be produced by two different species. For example, microcystin can be produced by microcystis and also anabina. So it can, yeah. So it is gene specific, not species specific. That is why morphological identification for cyanobacterial toxin is actually quite difficult, because you see the same morphology, one produces, the other one doesn't. So the answer is yes, you can, but you have to do molecular test. Of course, you can use chemical test, but there is a problem with chemical test. You see in the lake, some dye, and then you think it's toxin, cyanobacterial toxin, but then some of them, the half-life, half-life is, what's half-life? Fasa hidup, half-life, yeah. Some of them, the half-life is quite short. So they kill you with the toxin, and then they disappear because of degradation from the sun, the temperature. So you see someone died, but the toxin is no longer there. So that's why sometimes chemical analysis can be quite tricky. So the best way to do it is by gene. Yeah, detecting the specific gene encoding for that toxin. Thank you.
Okay, whose will be the next? Do you have any questions? Or maybe from the Zoom? Oh yeah, we have one here.
Okay, thank you for the opportunity. My name is Hani. So my question is, uh, because we talk about potential of cyanobacteria, I think the potential of cyanobacteria is really interesting. But I'm curious, would it be more feasible to harvest cyanobacteria directly from herphoom blooms, or just cultivate selected strains under controlled conditions? Thank you.
I don't understand. Mm. Okay, 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 okay. Oh, okay. Thank you very much. The answer is yes. We are actually looking for cyanobacteria from alami, from the environment, and then we isolate them. And we also have culture in the lab, which lasts for like many years. But the issue about the culture, cyanobacteria has morphological differences. They're going to change their morphology according to the condition. Like the filamentous length will be different according to temperature, according to the lights. I think cyanobacteria is very good, because I think the philosophy is not having everything that you want, but making use of what you have. So if they don't have a nitrogen, they're going to produce a lot of heterocysts to fix the nitrogen. Once they have the nitrogen, they don't produce it anymore. They just use the existing nitrogen. So yes, the answer is yes, you can get them from the environment. We also have culture in the lab. And cultural manipulation, you can. The genetic engineering, yes.
Bahasa Indonesia aja ya. Bisa ya kalau kita mau kulturkan, jadi diisolasi dari alam, kemudian dikultur di laboratorium, bisa dengan manipulasi kultur. Misalkan kita mau tingkatkan apa kandungan toksinnya atau kandungan apa, kita bisa manipulasi dengan menggunakan cahaya atau suhu atau apa saja. Itu namanya culture manipulation. Tapi kalau mau pakai untuk gen, misalkan dia nggak bisa dikultur di laboratorium, biasanya gennya yang di isolation, kemudian ditransgenkan ke dalam spesies yang mudah dikultur di laboratorium, itu yang akan nanti memperbanyak untuk bahan aktifnya. Gitu.
Exactly, Prof. Thank you so much, Prof. Elmi. Bagus juga ada bantuan yang sangat hebat ya. Ya, actually, for example, kalau untuk jadi skincare, kita nak produce a lot of pigment yang block skincare, kita letakkan UV. Kita letakkan banyak-banyak UV, so dia akan produce. Kalau tidak, without that UV, nanti dia doesn't produce the compound that we want. So, like Prof. Elmi says, we can actually do cultural manipulation. Thank you. I think I'm going to give you this, what is this, elephant seal. Thank you very much.
Thank you.
Thanks.
Okay. Do you have questions? Yes, from Bu Reni, silakan.
Makasih. Saya bahasa Indonesia saja ya, Pak.
Oke, saya tertarik dengan cyanobacteria ini. Kalau kami orang aquaculture, kami biasanya musuh besar cyanobacteria karena itu akan merusak hepatopankreas daripada udang, ya. Kemudian akan menimbulkan bau yang tidak sedap pada kultifan kami. Tetapi tadi saya dengar bahwa kalau nggak salah ya, dia sebagai apa? Pada mikroplastik. Dia sebagai substrat ya atau gimana? Iya, berarti
Breakdown, dipecahkan.
Iya.
Degradasi.
Degradasi ya. Berarti boleh kita memperbaiki lingkungan dengan cyanobacteria atau gimana? Tapi kan itu bisa juga belum ya, habis itu. Makasih.
I think I like your question because I think everything.
Sorry, bahasa Indonesia bisa?
Ya. Kalau
Melayu.
Malaysia, saya rasa
Malaysia, ya.
setiap perkara kita boleh melihat dari segi positif ataupun negatif. Sangat penting. Kalau kita ada, contohnya alga bloom dalam cyanobacteria, dia boleh jadi bahaya menghabiskan oksigen. Tapi kalau dia boleh produce oil, alga oil, kita mau yang bloom forming sebab dia boleh cepat membiak. Dan kalau kita mahu yang seperti mana udang dan dia akan mati, kalau kita nak udang itu mati, kita kasih makan. Kalau nak survive, jangan kasih makan. So I think apa-apa dalam hidup tidak akan ada sesuatu yang bagus untuk semua. Tertakluk kepada tujuan. Saya rasa purpose is sangat penting. So memang ada cyanobacteria yang berbahaya. Contohnya Sclerosis, dia ada produce toksin dan boleh bunuh. Jadi saya rasa Okey, untuk plastik, saya rasa pada tahap kami punya knowledge, cyanobacteria boleh ambil fosforus daripada plastik. Jadi dia akan menyebabkan plastik itu mudah terurai. Sebenarnya for the current stage, saya sangat pesimistik sebenarnya. Sebab saya rasa dia tidak ambil karbon daripada plastik. Saya tidak rasa dia akan breakdown sampai CO2, karbon dioksida. Dia hanya akan convert plastik into microplastic. That's my current opinion. Sebab itu saya tidak mau guna teknologi ini. Sebab saya rasa dia tidak akan. Jadi sebenarnya tujuan kita untuk pecahkan dengan plastik kepada CO2 dengan harapan the autotroph akan take out the phosphorus and heterotroph akan break down easy for heterotroph macam kulat untuk break down the carbon into CO2. Tapi for the current stage saya rasa hanya akan sampai ke microplastic, dia belum sampai ke CO2. So pada saya, kalau kita kata dia bagus untuk lingkungan, for the current stage, I don't think so. I think sebenarnya lagi tidak bagus. Tapi kita tahu dia capable. Yep.
Satu lagi, kami kan ada kegiatan kultur. Nah sekarang kan tahu bahwa suhu ini sangat tinggi, ya, evaporasi sangat tinggi, dan itu memicu cyanobacteria yang blooming di tambak kami. Kira-kira apa yang harus kami lakukan untuk mencegah itu, Pak? Karena kan semakin tinggi ya, bukan juga cukup ekstrim, tetapi dia semakin tinggi itu kecepatan pertumbuhan itu sangat cepat. Bagaimana menurut Bapak?
Saya rasa isu yang sama yang kami hadapi di United Kingdom masa saya buat S3 adalah alga bloom di drinking water reservoir, dan dia bloom dan sangat cepat, terutama kalau suhu naik kan dia akan lagi cepat, dan kalau lepas itu dia mengurai, dia akan anoksik ya sebab tidak ada oksigen lagi. Tapi that's why kami buat kajian tentang kami buat di UK, dia pakai barley straw. Straw is jerami bukan padi, tapi barley punya jerami untuk control. It's actually traditional method. Tapi sebab kita tidak ada jerami barley di Asia Tenggara, so that's why kami gunakan palm oil trunk. So sebenarnya kita boleh kontrol guna lignin derivative. Kalau if you ask me to recommend, saya akan masukkan batang kelapa sawit, kalau boleh di dalam, dan dia akan slowly rotten and release lignin and lignin derivative. Actually, there is a published paper published by Xiao et al. 2000, I think 15. The Salcolin A and Salcolin V can actually control cyanobacteria. I can give you the paper. Tapi I've never done it myself. All I did was lab scale for palm oil trunk. It can do for lab scale, but for big scale macam kultur saya tidak buat lagi, belum. Ya, tapi sebenarnya kalau kita tahu apa yang akan bunuh cyanobacteria, kita tahu yang penting sekali cahaya. Cahaya tak mau. Without lights dia mati, confirm. Sebab macam rumput pun kalau tak ada cahaya dia mati. Kecuali macam chlorococcum dia mixotrophic strains. That is tahan. Sebab kalau dia mixotrophic ini, kalau I think you know lah kan, tapi kita terangkan kepada student lain. Mixotrophic dia boleh buat fotosintesis dengan juga heterotroph, dia boleh macam makan dan dia boleh fotosintesis. So ini yang selalu kami guna sebab dia tahan bila tak ada cahaya dia hidup. Tapi I think the single biggest enemy of cyanobacteria is cahaya matahari. Tak ada dia akan mati. Macam kita cover, that's why di empangan-empangan di kebanyakan juga di negara maju diorang letak bola-bola hitam yang terapung di atas empangan dan tak ada masuk dan dia pun mati. Kalau lain-lain, kalau macam kadang-kadang tak ada pollution pun dia akan survive because like we said they can fix nitrogen. So tak ada limbah ke pollution, tak ada limbah. The composition of species is going to change. Kalau ada limbah yang tak boleh fix nitrogen akan membiak dengan cergas sama-sama dengan yang lain. Tapi kalau limbah tak ada air bersih pun letak di luar, yang dia can fix nitrogen. Yang penting ada seeds, biji benih satu, ada cahaya, memang hijau. Sebab dia boleh fix. So it's not necessary pollution. Kalau kita tengok dari segi sejarah, cyanobacteria ini berjuta-juta tahun lalu manusia tak ada lagi. Cyanobacteria sudah wujud. Tak ada kaitan dengan yes. Ya, belum ada kita lagi, dia sudah ada. Kita yang bahaya kepada dia. Dia bukan, ya. So the species composition is going to change according to the nutrient level, the light's intensity and availability of nutrients. Tapi memang akan ada di situ. Macam hutan kita pun kalau kita tak kacau, kawasan itu terus jadi hutan. So it's the same. Is that answering the question? I hope it does.
Terima kasih. Karena kan anabina memang memfiksasi
Yes.
nitrogen.
Ya, anabina heterosis dan boleh produce toksin lagi tuh, anatoksin A and microcystin.
Ya.
Bahaya anabina.
Ya, makasih.
Makasih.
Oke, terima kasih Bu Reni. Is there any questions? Yeah, Bu Reni, you have.
Doraemon,
You get
sorry. Lupa bagi Bu. Oh, terima kasih.
Yes, this will be last questions.
Okay.
Okay, previously, Prof said that cyanobacteria could act as a barrier to UV rays. My question is, what are the risks if we add cyanobacteria to skincare or cosmetic?
Thank you for the very tough question. So what we're saying is that for cyanobacteria, to be able to block the UV, they produce compound that stops UV from killing them. And that compound is called scytonermin. So when we add, we can actually use this same compound in our skincare product to actually block the sun ray from going into your skin. But we haven't gone into that clinical trial yet, so we don't know what will be the side effect. So the answer is we still don't know because we haven't gone through that. But I think that is a very valid concern because in the future, when we come into this stage, we need to test that. For now, we don't know. Is that answering your question?
Thank you.
Actually, saya tidak tahu jawabannya.
I mean, is this from the compound, from cyanobacteria, or directly from the species to protect as a UV, anti-UV? The compound from from bacteria of the compound. Schitonema. Yeah, name. Oh, okay. Thank you.
Actually, Prof. Elmi make a very good point. I didn't know where Scytonemin is coming from, but I think it probably first isolated from Scytonerma, which is a species, but I didn't know. Yeah, I suspect it is. Yeah. But Scytonemin is the compound which is used by cyanobacteria to protect themselves from UV radiation. And it's colorless. There is no color. So it's very good when you put into your skin. You don't really see any color, but it can actually block the UV up to 99%. This is like 90%, 99. It's actually quite high, yeah. Yeah. But I only use that picture of UV50, but according to the literature, it's about 90%. I think even, 90, which is quite a lot. Yeah.
Itu compoundnya itu senyawanya namanya skitonim kalau nggak salah. Tapi itu berasal awalnya dari skitonema, salah satu jenis cyanobacteria. Dan itu jadi semua itu bisa didapat dari kenapa tadi dia mungkin dalam bahasa Inggris ya, dalam bahasa Indonesia. Sebelum kita ada di bumi ini, makhluk hidup, yang pertama ada di bumi adalah cyanobacteria. Kenapa cyanobacteria bisa ada pertama kali di bumi? Karena cyanobacteria itu tidak butuh oksigen. Justru dia menghasilkan oksigen. Nah, semua makhluk hidup waktu itu tidak ada yang bisa hidup karena menunggu ada oksigen dulu. Siapa yang mengadakan oksigen di bumi ini pertama kali? Itu adalah cyanobacteria. Nah, jadi dia itu ada 3,8 miliar tahun yang lalu, miliar ya, billion. Jadi itu ketika dia mengadakan oksigen di muka bumi, semua makhluk hidup mulai tumbuh satu persatu. Mulai dari bakteri, mikroorganisme, kemudian makro dan tumbuhan, hewan, akhirnya sampai ke manusia. Jadi disitulah kelebihan cyanobacteria untuk dimanfaatkan dalam berbagai aktivitasnya. Tadi contohnya dia mengambil sampel cyanobacteria dari Antartika. Jadi cyanobacteria itu ada di semua habitat, mulai dari from the Arctic and sampai ke gurun Sahara, kemudian sampai di daerah apalagi yang itu di Leca atau sini mungkin ada yang asal dari Leca. Itu sumber air panasnya itu kalau Anda ambil sampel akan ada Anda temukan cyanobacteria di situ yang tahan terhadap suhu panas sampai mendidih. Makanya itu banyak digunakan sebagai PCR enzim PCR karena di PCR itu kan harus enzim yang digunakan itu harus tahan di atas 100 derajat Celcius. Cyanobacteria bisa menghasilkan enzim-enzim seperti itu. Jadi itu banyak sekali riset sekarang mengarah ke cyanobacteria karena dia tahan hidup di kondisi ekstrim. Ekstrim suhu, ekstrim cahaya, ekstrim tekanan, ekstrim ekstrim again? pH, dan semua ekstrim. Jadi itu menarik nanti kalau ada yang mau penelitian tentang cyanobacteria di laboratorium itu bisa dimanipulasi. Anda bisa mengasih dia sebagai medianya adalah air panas. Itu bisa, Anda bisa dapatkan spesies-spesies yang tahan seperti itu. Saya kira itu aja tambahan dalam bahasa Indonesia ya. Sorry, my English is so many mixed with German and French. Okay, thank you so much.
Thank you, Dr. Helmi. And will be end our session. And once again, let's give applause for Dr. Jap for the very insightful presentations and discussion, and for us as well. And that's all for our...
Can I give some present to you all? I also brought some for you all.
Oh,
yeah,
yes.
But maybe after this.
After
yeah.
this,
Yes.
Thank you in advance. Okay, so that's all for today. I hope this will give everyone insightful based on this presentation and discussion. And hopefully, maybe if you have more questions, you can contact with us or maybe later with Dr. Jeb himself. And if you have any interest for the internship, maybe we will build the networking for you. So that's all for today. Thank you very much. I will give it back to the.
Jadi kita rencana juga kolaborasi ya, kolaborasi riset atau apa. Kita juga rencana fakultas mungkin akan mengadakan kayak kolaborasi riset, dan kalau anda mau ada penelitian, mau ini kebetulan kalau ada kerjasama proyeknya bisa ikut ya. Jadi itu juga untuk bisa memperluas cakrawala ilmu dan penelitian Anda ya. Saya kira itu anunya bagus juga. Oke, thank you so much. Baca doa kali perlu. Ya, dengan mengucapkan alhamdulillah kita tutup acara general lecture hari ini. Semoga ilmunya bermanfaat dan thank you untuk Prof. Jab. Semoga jangan bosan-bosan ya Prof untuk datang selalu kapan-kapan kalau ada apa event-event bisa dan kita mungkin juga bisa berkunjung ke sana Anda ada inbound dan outbound. Bisa nanti ke universitas beliau. Jadi universitas beliau itu menerima juga untuk magang atau apa, nanti kita akan bicarakan. Mudah-mudahan itu bisa membawa sesama negara ASEAN ya menjadi lebih lebih akrab untuk saling bertukar ilmu. Saya kira itu. Assalamualaikum warahmatullahi wabarakatuh. Mungkin Bu Uni mau ada kata-kata nggak? Bu Uni ada mau